It's a great pleasure to introduce Dr. Bruce Logan from Penn State. He is a professor in environmental engineering and at Penn State, as well as the director of the Environmental Engineering and Environmental Institute, as well as the Hydrogen Engineering Center. He's also an investigator at KAUST in Saudi Arabia for a number of years. He's holding 2 endowed professorships, one from the university, one from the department. But what is really outstanding is his contributions over decades by exploiting how actually a simple observation that microbes are able to electrically interact with the environment, how this simple idea he took and really developed the science and technology that ultimately there's a new way to use microbes for water treatment, for freshwater and for wastewater treatment, as well as for bioenergies. And I think it's fair to say that he single-handedly developed this field and is the undisputed leader in this aspect. He's also a GSEP principal investigator, has authored more than 340 publications, including books that are sort of the standard of the field, and has also received numerous awards, including the Clark Water Prize reward in 2009. I'm especially delighted that you beat the adversities of the nowadays air travel, which got him into the Bay Area at 2:30 this morning. So thanks for coming, Bruce.
Thanks for that very nice introduction. And now it's also nice to know that if I make a slip up anywhere, I have a good excuse. So I'd like to be talking about some things which are very much different, probably, than what you've heard before. And I'd like to introduce this as sort of a whole new platform for how to capture energy in ways, and in many cases, remarkable ways that we're not currently doing. Now, to do this, you have to develop new technologies. These are water-based technologies, and every new revolutionary idea goes through 3 stages. It can't be done. It's possible, but it's not worth doing, probably where we are now, and hopefully, I said it was a good idea all along. And we see that in water, different types of water devices, reverse osmosis membranes for water treatment, membrane bioreactors for wastewater treatment. These are technologies that could not be done. And so perhaps these microbial electrochemical technologies fall along the same line. People have been saying can't be done, but maybe it can be. And so these technologies, at least for my talk today, will fall into 2 broad areas: microbial electrochemical technologies, primarily microbial fuel cells, and then how to make methane from this energy. And time permitting, I'll talk a little bit about some other ways to capture energy using these very same platform technologies. And to give you a scope, people may not be experts in biology or water technologies, but to give you a scope of the kind of energy we're talking about here, we use about 17 gigawatts of energy, continuous power in the U.S. For just our water infrastructure. And yet that same wastewater contains about 15 gigawatts of energy, of power. So if we could save that 15 gigawatts that we're using, that's a 3-fold magnification of primary energy, and then capture that 17 gigawatts, that's suddenly the creation of about 60 gigawatts of power. We also have available from these technologies novel ways to capture energy in cellulose. I'm going to talk about the potential to capture energy salinity gradients, both natural and artificial. And those artificial salinity gradients will be a method of capturing waste heat energy. So this is a microbial fuel cell. This is the basis of this. It's just a box with bacteria. Those bacteria create an electrical current which powers this fan. And this is actually on a webcam. We show this because it's just, it always seems too unbelievable that without any moving parts within that reactor, the bacteria could create this electrical current that would be capable of powering this fan. And we've had several of these devices operating over the years. And how it works is the bacteria degrade organic matter and they release those electrons to a surface. those electrons have to flow to the counter electrode because there's a thermodynamic potential where those electrons combine with protons in the water and oxygen to form harmless water. The devices that we use to do this are really quite simple. They're just two electrodes, one exposed to the water and the other one exposed to the air and the water. And we can take a little piece of plastic, drill a hole in it, put the electrodes on either side, and we can make electricity. Now, the way that bacteria get energy and the way that we get energy from this process is when we eat food, what we're really doing is oxidizing organic matter. We're removing electrons. Those electrons are carried to another set of enzymes in our body, and those electrons, when they're transferred through respiratory enzymes, actually is what creates our ATP, our energy source. And so respiration is really the key for creating the power. It's not the eating, it's if you will, the breathing that allows us to make this energy. The ability of bacteria to do this, to release electrons outside the cell and to accept electrons into the cell, has created a new form of microbiology, which we are calling electromicrobiology. And if you go into my lab, it may not look like a microbiology lab. It probably looks more like an electrochemistry lab because of all the meters and dials and bottles and so forth. But it really is all about the microbes and about the structures that we build around these microbes. So we can think about these microbes as being very special in two ways. Either there are electrogens, they can release electrons out of the cell and to an electrode, or electrotrophs, microbes able to take those electrons into the cell and to use them and to make electricity as well. First, let's talk about the electrogens. We take an electrode and we expose it to a natural water sample. Bacteria that can respire with that electrode grow very quickly and colonize the electrode, and then they make electrical current. And they can do that in different ways, but probably the most fascinating way that they can do that is by making self-made nanowires. They actually make their own conductive pili to conduct those electrons from inside the cell to a surface. They can do that other ways in addition to nanowires. For example, they can produce little electron shuttles that will carry those electrons from the cell to the surface. Some microbes have to make direct contact, and other ones are sometimes there just kind of living off the activities of all the microbes in the biofilm. Now, the electrotrophs are capable of now capturing electrons into the cell, and a couple examples of that are, for example, for oxygen reduction. There can be bacteria on the other electrode that facilitate the capture of the electrons from the electrode and releasing those to oxygen. Some microbes can use nitrate to release those electrons too. Other microbes, metals, and probably the most important one for my talk here is CO2 reduction by methanogens. So the evidence that we have for this is often that when you put more nitrate into the system, that's not working very well, but when you put more nitrate into the system, that you see an increased amount of power production in the system. That's a good example of the bacteria using that nitrate, because if they didn't do it, the nitrate wouldn't have any effect. Other microbes can catalyze oxygen reduction, so reduce the overpotential of the electrodes and allow greater power to be generated as well. In the case of CO2 reduction by methanogens, it's been known for a very long time that certain microbes can break down organic matter, primarily ferment that organic matter to release, for example, acetic acid and hydrogen. And the traditional paradigm is that the methanogens would then use that hydrogen and fix CO2 and create methane. But along the way, we discovered something else quite interesting, because what you have in this existing paradigm is a very fast reaction by one microbe and a very fast reaction by another microbe, hooked up by a very slow reaction, the diffusion of hydrogen from one microbe to another. Why waste all that time? It's like building a dirt road in between two superhighways, which creates a lot of traffic jams, which we all know about coming here today. And so why have that slow road? Why not just have those two microbes directly contact each other and have those electrons directly transferred? Well, that indeed does occur in nature. We've shown this through seeing the presence of these nanowires. We've shown it through mixed and pure cultures as well as through methane generation. So the first evidence of this in the that we showed in the literature was a fermentative microbe, something capable of breaking down sugars, connected to a methanogen by an electrically conductive appendage. That wasn't proof that it was occurring, but it laid the foundation for the possibility of this to occur. It wasn't until we actually took a culture and put it onto the cathode where the electrons are being delivered by a potentiostat in the lab, and we saw that we could get methane release as well as CO2 reduction with no other source of electrons in the system. And so, for example, if you were to look that lower line, that's how much hydrogen could be coming off the system that would produce methane. And you can see those green lines showing much greater evolution of methane than would be possible directly from hydrogen. We were also able to show that you could get this, that these current densities were much greater than you could get by hydrogen alone. So, since we did that work, there was some other evidence now that's emerged in the literature, and it's on electrically conductive granules and anaerobic digesters, basically showing that there are conductive surfaces within these granules that produce methane, and that these microbes must be doing this naturally in existing systems. There's also some work that shows when you put in additionally electrically conductive material into the system that you promote the rate of methane generation. And so people have looked at, have known for many years that activated carbon being added to an anaerobic digester increased methane, but they didn't know why. So this new work has added to the literature showing that this can occur. And to have a little fun for a minute, this is a good example of it can't be done. There was a paper produced by Derek Lovely's group which basically said we tried to do those experiments that Logan did and we couldn't reproduce them. It can't be done. And then they produced this paper I was talking about. Actually, this is Derek Lovely's group, and it said, for the first time, we've shown that direct electron can occur. So maybe the way the saying ought to go is, it can't be done, it's possible, and it was my idea all along. Sorry, I can't resist sometimes. So we know these things are occurring, but we don't know how they're occurring, and that, of course, is where the really interesting science comes in. We assume that these electrogens are making the nanowires, but we don't know how specific this interaction is with the electrotroph. Is this like, like an enzyme thing, a plug and a socket relationship? But not all plugs look like that. You go to the UK, they look like that. You go to Europe, they look like that. And those aren't going to fit into those sockets. Or maybe there are universal receptors for universal plugs, or maybe there's universal donors. We don't know how this works. We don't know how much other microbes can make use of these pathways. And that's where all the fun will be ultimately. So the basis of my project here, which is just getting underway, is to really look at this ability of these methanogens to turn an electrical current into methane. We can do that electrochemically without any microbes, so just an abiotic reaction, but it usually requires precious metals. You get a lot more than just methane for your money, and you don't really want that. You get a lot of side products. And yet you put these methanogens on there, and they're, shall we say, not very smart. They only know how to do one thing: make methane. But that's precisely what we want: a nice, clean, and pure biofill from that electrical current. So how do you do that? Well, take a system that you have making electricity using a microbial fuel cell, and then you take away the oxygen, and you know what happens now? Nothing. Well, that's not very good. So what you need to do is you add a little bit of power to the system, you boost the voltage, and now you can drive spontaneous hydrogen evolution from that other reaction. And the advantage of this system, let me go back there, the advantage of this system is you're only using about 1/10 of the voltage you need to split water, because the microbes are actually splitting the organic matter. You're not doing that energy-intensive process of splitting water. Take that system then, and even if you don't have anything on the anode, organic matter, so you would have to split water in this case, But if you put your methanogens on the cathode and you use a source of electrical power to create that current, you can turn any source of electrical current into pure methane. So in addition to making methane in anaerobic digesters, the way that we do this now, you could also make methane using solar energy or wind energy or other forms of energy. So the goals of this project are to identify the methanogens that will work best in these systems, look at different catalysts that can help us reduce the overpotential, that is the voltage that we need to apply to the system, so that we can get the most efficient conversion of this electrical power into methane. There are two basic types of methanogens, the ones which I've mostly been talking about called hydrogenotrophic methanogens. The other ones are acetoclastic. They use something like acetate or other types of organic molecules. We're most interested in these hydrogenotrophic methanogens. And the total voltage that you need to apply the system occurs from two things. It occurs from the reaction occurring at the anode and the reaction occurring at the cathode. And if you have a biotic anode, then you have 0.3 volts being actually produced by microbes. If you have to split water, you're going to need to input minus 0.8 volts. So obviously there's a real benefit of having microbes and organic matter on the anode. But what we want to do is to get the most efficient system on the cathode, and actually direct electron transfer requires about 1 volt of added voltage, and that's a lot. So by using a hydrogen catalyst, we can actually evolve hydrogen at much lower overpotentials. And a couple of good alternatives to the traditional catalyst, which is platinum, is stainless steel and molybdenum disulfide. Stainless steel is readily available, it's very inexpensive, but it has relatively large overpotentials. Molybdenum disulfide is actually a nice alternative, and it's being researched here at Stanford as well as other places, and this shows great promise as a very low overpotential catalyst. And what I mean by that is you can see that arrow shows that you really don't get much methane on it from a cathode until you hit minus 0.1 volts. But using a catalyst like platinum reduces the amount of voltage that you have to apply, and the higher loadings of molybdenum disulfide will reduce both the point at which hydrogen starts to evolve as well as giving us a good rate. And I know this is not, you're not a very specific technical audience, so just trust me on this, and it works. And we've compared platinum and molybdenum disulfide in these systems, and it doesn't go as fast, but it goes quite well. So really, we're just getting underway in the tests of making these different types of electrodes with different types of inocula and so forth, and maybe sometime I'll have time to talk about that. In order for these systems to become practical, to move from the laboratory into practice, we're going to have to scale up these systems. So let me just tell you about a few things that we've done to scale up microbial fuel cells and microbial electrolysis cells. This is a 2-liter laboratory demonstration project that I showed you right up in the beginning, spinning that fan. And it's actually the biggest microbial fuel cell that we built. We build smaller ones because we want to focus on the aspects of scaling up, but we know how to scale these things up. We need to make these in modular forms with very closely spaced electrode, and then we need to pack all these modules into compact reactors. These reactors won't be the kind of reactor that will fit on the back of your car and power you down the street. They're going to be much larger, but they will be of the size that are typically used, for example, in wastewater treatment. And so with a microbial fuel cell, what you want to do is have a chamber where the bacteria can attach to electrodes and water can flow through that channel, and then you have another inner channel where air can flow, and that's a pretty complicated system to develop. Now, microbial electrolysis cell is actually much simpler. You basically need to put 2 electrodes into the water. And this is a reactor we developed. It's about the size of a shoebox. It's 2.5 liters. It's got graphite fiber brush anodes and stainless steel cathodes, so no precious metals. And we inoculated the reactor and got it running, and it did very well. All these lines, each one of these lines is the amount of current being generated from each one of those anodes. And you could see a pretty good amount of consistency between the electrodes. The problem was this reactor was actually designed to produce recoverable hydrogen, and it didn't. In fact, this was our first microbial methane generation system because it beautifully moved from all hydrogen to all methane. So it turns out we do know how to build a methane reactor already, maybe just not the most optimal one. So we took this idea and we decided we'd try it at larger scale to see if we could predict how well we could go from the lab to the field. So we had to go somewhere in the world to do this, and so you know, I'm in Pennsylvania, that's where my lab is, so of course I went to Napa, California. Just call it a hobby and work combined. We went up to the Napa Wine Company and we built a 1,000-liter reactor with 24 modules with a total of 144 electrode pairs. This system was about the size of a bathtub or a refrigerator on its side, and we predicted what each module could produce, and that yellow line is the current from each module that we predicted. And you can see we did pretty well. We had to eventually stop the test because the winery's wastewater production occurs only over about 6 months, but we felt very confident that we could scale this system up. Now, let's see, how much time do I have left here? I was a little, my time slot is officially up, but I started 10 minutes late, so I have about 7 or 8 minutes. Okay, good. So let me just spend just a few more minutes talking about other ways to capture energy, because I've just told you we can make methane, but we need to use electrical power to do that. We might not want to do that. So where else can we find that energy? Well, one place is salinity gradient energy. The other one is waste heat energy. Let's talk about salinity gradient energy first. Getting more energy. Nice picture of an ocean. We'd all like to perhaps be at that ocean now. But for every area that has an estuary, and most estuaries are developed, you have fresh water flowing into salt water. Now, you don't realize it, but there's a huge amount of energy in that system. And if I were to translate that energy into something probably more easily grasped than thermodynamic numbers, it would be that the energy difference is equivalent to water flowing over a dam 270 meters tall. This is every time wastewater goes into the ocean, or every time river water goes into the ocean, that's the energy difference that exists that we could be capturing. And that could be, some people estimate, as much as a terawatt of energy around the world. Now, how can we capture that energy? Well, to explain that, I first have to explain electrodialysis. And electrodialysis is where you take two electrodes, an anode and a cathode, you apply a potential between those electrodes, and then you have alternately sequenced anion and cation exchange membranes. Anion exchange membranes allow negative charge, like chloride. Cation exchange allow positive charge, like sodium. And so you create more concentrated and more dilute solutions, and this is a way to desalinate water. It takes a lot of energy. But just like running uphill takes energy, if you're on top of the hill and you go down, you can gain energy. So in reverse electrodialysis, what we do is we introduce a salt water, seawater, in one chamber and a low saline water in the other chamber, like river water. Now, anions can only move through one membrane and cations can only move through the other membrane. And so what this does is it creates electron flow. It creates a thermodynamic potential, which is allowed by electron flow between those two electrodes, where each of these pairs contributes about 0.1 to 0.2 volts. And so like linking a battery with other batteries in series, putting in a whole series of these membranes adds up to create more and more voltage. Now, this has been known, but what we realized was that the electrode reduction was really ruining our efficiency for capturing that energy. So we actually put one of these reverse electrodialysis stacks into our microbial fuel cell. And what we found was we got greatly enhanced power. We increased the amount of voltage we produce as threefold and the amount of power about tenfold. Well, that tells us how we might capture salinity gradient energy, but what about waste heat energy? Here, the idea is we take a specifically constructed solution, for example, ammonium bicarbonate, and we apply very low-grade waste heat to that solution. And what happens is, like alcohol distilling out of water, but we don't need to boil the water, the ammonium bicarbonate volatilizes out, leaving a low concentrate solution behind. We condense that ammonium bicarbonate out. We now have our high concentrate solution. So we've taken that waste heat and distilled it into a low and a high concentrate solution. That is our artificial salinity gradient. And if fresh water and seawater is 270 meters, this is something closer to 400 meters in terms of water flowing over a dam. So how does this all help us in making methane or making hydrogen? Well, if you go back and you put that stack of membranes into what we call this microbial electrodialysis cell, remember the one that needed extra power to make hydrogen? Well, this stack is the power source. We don't need any electrical grid energy, and we can use that salinity gradient energy to very effectively produce hydrogen at the cathode. So this is a system by which we could biotically make hydrogen in a completely new way to make methane, or we can do it abiotically as well. We just need more membranes there to split water and evolve hydrogen at the cathode. So I'd like to conclude there. I don't even know how I put together a summary slide from all that, but I'd like to end by thanking my group, which is an extremely diverse group of students because there are electrochemists, microbiologists, engineers, material scientists, and so forth. And I'm grateful for the funding through KAUST, Air Products, the National Renewable Energy Lab, ARPA-E, DOE, a new project by the DoD, and of course this project through GSEP. And if you'd like any other information, I'd be happy to refer you to my website and other venues such as Twitter and YouTube. And thank you very much for listening.
Thank you, Bruce. Thanks for this very exciting talk. I think we have time for a few questions before the break. Yes, please.
I don't think, maybe I can speak louder.
Hi. I was interested in your first set of slides where you actually showed nitrate getting converted. And today, to purify water and removing nitrate is a very expensive proposition.
Yes.
So could you actually utilize your technique to do that?
Absolutely. Yes, you could. You could also, there are many different ways to get nitrate out of water. The question is, could you do it economically? But absolutely, you could do it this way.
I think everybody is ready for a coffee break.
Okay.
Bruce will be here as well as the other speakers. Please thank all speakers for this morning for a great session on bioenergy.